So this is Andy Pekosz, Associate Professor of Microbiology and Immunology at the Johns Hopkins University Bloomberg School of Public Health.
And this is Brian Simpson, editor of Johns Hopkins Public Health magazine. We're here today to talk about the H1N1 Influenza virus. And Dr. Pekosz, thanks very much for talking with us today.
My pleasure.
Let's begin. The H1N1 virus that caused a pandemic this spring doesn't appear to be as lethal as a regular seasonal flu. So why are people so concerned about it?
Really, there are three things that make us concerned about this new H1N1 virus. One is that its surface antigens are different from those present in current human influenza virus strains, which means that there's very little pre-existing immunity in the human population. It is also spreading at a time when human influenza virus usually doesn't spread in the human population. So that's a little bit interesting and worrisome to us. Finally, it's a new virus in a new host. So there will be a lot of selective pressure on this virus to adapt to replicate in humans. And this may have implications for its ability to cause disease. It may increase disease, it may decrease disease. We're just not quite sure.
And is there any predictability about that, sort of those mutations that does it tend to get worse or does it tend to get better for humans at all?
Well, we know very little about how influenza viruses transmit from person to person, other than the fact that coughing and aerosols and surfaces that are in contact with the virus can spread the disease. Oftentimes, viruses become less severe as time goes on, but certainly there are examples from other influenza pandemics of viruses which entered the human population in a rather mild form and then became more severe upon subsequent transmission through humans.
Mm-hmm. And what is it about a flu, that virus, that makes them so dangerous?
Well, influenza infects the respiratory tract, and in fact it can infect all the cells that line your respiratory tract, from your nasal epithelial all the way down through your lower lung. So therefore it makes most individuals very ill with these kind of respiratory problems. Of course, that means it's especially dangerous to people who have pre-existing respiratory problems or who have weak immune system. Particularly the very young, the elderly, and pregnant women.
Mm-hmm. Mm-hmm. And tell us about the H's and N's that we hear about in H1N1 or H5N1. What do those mean, actually?
So we group influenza viruses together based on how similar their surface proteins are, because it's your antibody response to these proteins that protects you from infection. An H1, for instance, is very different from an H5. Use the analogy of colors here. H1 is red, whereas H5 is blue. If your immune system recognizes one, it will not recognize the other because it is so different from the other one. With the new 2009 H1N1, it's a bit more subtle. It's more along the lines of color shades. If the seasonal H1 is a dark red, then the new H1N1 would be pink. They're closer. Most people could tell you that they're related, but certainly the colors are very different from each other, as they are from the blue of an H5 virus.
Mm-hmm. And so if I've been exposed to an H1N1, that doesn't mean that I'll have immunity or any particular immunity to another H1N1. Is that right?
That's correct. Usually the best immunity is to the exact same strain that you've been either vaccinated with or exposed to. So if you've been vaccinated, you have a fairly good chance of being protected from that particular strain. If the virus strain changes a bit, then you wouldn't be protected from infection with that virus.
Mm-hmm. And I guess it seems like the key thing about influenza viruses is that they do mutate so frequently. Why is that?
Well, the enzymes that replicate the RNA that make up influenza genes make many mistakes. They also replicate the viral genome very proficiently. So within 9 to 12 hours of an infection, you can have hundreds of thousands of copies of each of the genes. So this combination of making mistakes and very high replication rates means that the virus is throwing out lots of gene segments with lots of mutations. Now, most importantly, the two surface proteins of influenza can tolerate these mutations much more readily than many other surface proteins can. So you've got the situation where the virus makes mutations in its genome. The proteins on the surface can tolerate those mutations, and therefore you can get lots of different variants of influenza viruses being thrown out from an individual.
Mm-hmm. And so then we're really faced with sort of natural selection going on, so that the viruses that adapt the best to with these mutations, these strains that have the best mutations will survive and really propagate in humans. Is that right?
Exactly. And where that's most important is in this ability to evade the pre-existing immune response. If an influenza virus with a particular change in the H protein, for instance, that causes it to lose a reactivity with antibodies emerges, well, that virus probably has a more greater chance of infecting the next person because that person's immunity won't be as great to that virus. And therefore, this idea that we have a pre-existing immunity is good because it protects us from disease, but it also applies a pressure to the virus to change to try to evade that immunity.
Mm-hmm. Mm-hmm. Great. And with all the gene swapping that goes on in influenza viruses, is there really much difference between swine flu viruses and avian flu viruses? Are some more dangerous than others?
Well, swine and avian influenza are both influenza viruses, of course, but they have very, very different diseases in humans. The avian viruses are much more severe than the swine influenza viruses. And we think this is because the swine virus lacks several key mutations that the avian viruses have, which confer on those viruses the ability to cause greater disease. So certainly the swine viruses can circulate in humans much more efficiently than the avian viruses, but they lack those key genes that are responsible for the very severe disease that we see with avian influenza virus infection of humans.
Okay. Dr. Pekosz, thanks very much for talking with us today.
Not a problem at all. Thank you.
Thank you. And we'll be talking with Dr. Pekosz next about past flu epidemics, the 1918 flu, 1957 and 1968 flus, and how the H1N1 outbreak this year compares with those.